Look, I got a couple things I'm going to be moving around. So, are you ready?
It's recording.
All right, so the first thing, let's know what we're playing with here. We have our double-stranded DNA molecule. It's got a left and a right side. You should view it as complementary strands. They run in opposite directions. We'll add some detail later about talking about the 5' and 3'. Don't worry about that for today. So you can just hold that right there. All right, so that's the players in this little activity. We also have, what does this represent? Messenger RNA. If you were to hold them up, all right, if you can hold that. All right, so number one, I'm holding it in the opposite direction. It's upside down for the camera view because now, although I didn't actually transcribe this, I didn't match this up, but imagine this. It looks just like the right side. Now I'm going to show the smaller ones to show transcription, but that's really what's going on. Okay? So there's our DNA. Now let's talk more about DNA. Actually, let's introduce all the pieces here. So you can just set that down for a second. So we got messenger RNA. There are 3 types of RNA, right? Yes. Messenger RNA, which we're most familiar with. When you do transcription, you're talking about messenger RNA. Transfer RNA, once it's produced, it goes to the cytoplasm and pretty much stays there. All right? And then there's this other one, ribosomal one, ribosomal RNA. Okay? So those are the big 3. And why should you care? Because this is how you get gene expression. What do we mean by saying gene expression? The whole goal is to understand to go from how do you go from DNA to proteins? That's what you really want to grasp. So first, I said messenger RNA, so I need to introduce what the other ones are. Here's tRNA, and I really don't have a model so this is going to represent transfer RNA, and you can tell when a transfer RNA and a messenger RNA connect, they connect through base pairing. Base pairing. So if it's A for messenger RNA, what would the transfer RNA be? U. Because it's RNA to RNA. Now, what you'll see is this is going to represent the amino acids here, and here's a long amino acid chain. Okay, so we got all of our players. The one thing I don't have is a ribosome, so I'm going to use my hands to represent a ribosome. And then let's talk about this transfer RNA because it's a little misleading. Look at this picture here. A messenger RNA is pretty obvious, a long strand. What is a transfer RNA? Now, I'm modifying this. So what it is about 70 to 80 nucleotides long. And what it does is, this is really what the transfer RNA does. It folds, twists to form a structure, and then it's got 3 parts up here that actually code, meaning they bond through hydrogen bonding with the messenger RNA, and then the amino acid will be attached to this side. So this picture doesn't do it justice. There's this part, there's a nitrogen base bonding side, and then there's an attachment point for the amino acids. Does that make sense? Yes, no, maybe. All right. So this is really what it looks like. I'm just you don't really do it justice with the models. Okay. So let's look at this, and let's go from DNA to protein. But we can quickly review replication of DNA. So remember, DNA is replicated, or DNA is what? Transcribed. Okay? So first, let's get a better understanding. Half your DNA, I shouldn't say half, I'd say about 95% of the time the DNA is not actually being replicated or expressed. What expressed means is DNA producing a gene turning on. What's a gene? It's a segment of a DNA that codes for a protein. So that's what we want to do. But let's look at this. Kind of tell me getting this here. So I'm not going to make you know enzymes today, but we will know it later. So here's the thing: is this DNA? See how it's all coiled up? And then what happens is, if I can add a prop here, let's say this is a protein. It's called a histone, and what it does is it wraps around it, and then say another one over here, another protein wraps around it. So this tightens up. Can you actually, do you have physical access to the nucleotides when it's like this? No. So what you have to do is you have to unravel it. And there's an enzyme. The first enzyme to know is one called helicase. What helicase does is this. It unravels the DNA. Okay? It unravels the DNA, and then there's another one that actually helps. You have some single stranded proteins that actually hold it up. So let's show how we get this DNA unzipped, because it does it for replication and it does it for transcription. Alright, so this is just for, it's easier for me to work like this. So it was coiled around, it was twisted, the twisted ladder. So my top hand is an enzyme called helicase that does this. Alright? The hydrogen bonds are broken. Now you have access. You can add more nucleotides here. You can add more nucleotides here. You can do this. Drop it on the ground. No, you can do this. Technical difficulties. Okay. So what happens when it's replication? This opens up and you get a new side, free nucleotides. It's actually one at a time. Gentlemen over there, I said we're taping this, so be quiet and listen. And then, look at this. We got two. The left side gets a new right, the right side gets a new left. We should better call it complementary strands. So, and now it doesn't happen like this, magic, but really, one side makes a copy. Now imagine this. If I were to hold this here, that'd be a centromere. This would be that doubled chromosome that you see during mitosis, because this would become one chromosome, this would be the other. So it doubles, and then you separate during anaphase. Does that make sense? All right, so that's one thing DNA does, is it replicates itself. The other thing it does is it makes RNA. So let's do that. I'm going to take, did I give you my open one? Okay. So I need a free RNA, which I have right here. So now, when this opens up, this time let's ignore that. A free nucleotide of RNA comes in. Now, once the phosphoester diester bond forms between the sugars and the phosphate, the nucleotides here, hydrogen bonds, then it breaks, zips back up. Now we have a messenger RNA that can go and produce a protein, or make a protein possible. So let's get to that stage, but where's the messenger RNA give you? The long strip. Okay. So what I want you to envision, we're in the nucleus now. This table is the nucleus. These guys are going to be the ribosome. So here's the DNA. It's doing its thing. It's doing its thing. It unravels, and then it does it does transcription, and now we have a messenger RNA. And this messenger RNA, so let's imagine this. This is a nuclear pore for the messenger RNA to leave the nucleus. So it leaves the nucleus. Does that make sense? So now where am I floating around in?
Cytoplasm.
Cytoplasm. So these guys are going to be a ribosome. I'm going to show it twice. Alright, so now, what you don't know, or what you wouldn't really see, is you've got transfer RNA is going to come in, and it's going to attach, and then you're going to have another transfer RNA and attach. So imagine two of them being like this. Okay? You have two transfer RNAs. And as this moves down, one of them is going to drop off, and another one's going to come in. So it's usually another one comes in, but what happens is this one over here is going to detach. So just to show you this, so that's kind of what's going on here. Now watch my hands. My hands are the ribosomes, and inside there is the tRNA and the mRNA, 3 nitrogen bases at a time. By the way, what is that tRNA called? No, that's the same thing. What's the tRNA called? Every anticodon, every 3 letters of messenger RNA would be a codon. So imagine, so you're zooming in, you can't really see what's going on, but as my hand moves down, you keep building and building and building and building this amino acid chain. Did that visual help any? Alright? As this goes down, alright, so my hands are the ribosome, and what's happening is transfer RNAs are coming in. If you were to zoom in, you would see this happening, the 3 bases at a time. And so there's one, and then another one would come in. That's why I have this. So as it moves down, just imagine 2 of them, but the top part where the amino acid is going is here. So my hands are the ribosome. As I go down this transfer RNA, I am translating and producing an amino acid chain. When we're done, there should be a stop codon here. That means the ribosomes actually separate these two parts. You have a free chain of amino acids now, and what it does is it forms a functional protein. If this was like the proteins like in muscle fibers, like, you know, I need a lot more to show the guns here, though. Now, what happens to this? We'll close by this. What happens to this? Hang on to it. As long as there are amino acids available and ribosomes, they're going to keep producing amino acids. This is how your body can express and produce thousands of genes, express thousands of product like in a couple seconds. Insulin does this. It mass produces. So imagine a ribosome coming down and that strip of amino acid. Then another one comes down. And, and every time this is happening, you're getting a product. And it's just it's coming down. And then when you're no longer you no longer need the molecule, right? Watch this. I'm not going to do it, but so as this messenger RNA goes back to the nucleus, as you're going through the pores, cut, it gets cut into free nucleotides of RNA so that it can be transcribed. It can be ready materials the next time another gene needs to transcribe something. It's the ultimate renewable resource. Question.
Okay, so we've talked a lot about like tRNA and mRNA.
Right.
What about like rRNA?
What is rRNA?
Yeah.
My hands, the ribosomes. rRNA is the ribosome.
Okay.
Okay? Those are the three. It takes all three. Really, what our ribosome serves as, it serves as an enzyme. Remember, enzymes catalyze reactions? But they do it at a faster rate, so you can produce a product when you need it rather than letting it. If those amino acids just float around and eventually would bump into each other and connect, by the way, every one of the bonds between the amino acid, we call that a peptide bond. You remember back from that? What this allows is for the proximity, the amino acid, one amino acid to come into contact with another amino acid, and then that causes the peptide bond. What holds them in place? The hydrogen bond between the anticodon and codon, the one that was most missed on the quiz. All right? And so that's what we need to make sure we're clear with. So that's how you go from DNA to proteins. If you don't understand that, that's half the battle for the future test. The only thing we'll add in is enzymes. What's a polymer? Something, one molecule, a bunch of repeating monomers.
Yeah.
So this would be a monomer. This would be a polymer. Okay. There's enzymes end in N, like pepsin, and ACE, like helicase. The two enzymes we're going to learn later is called polymerase. When I was showing a free nucleotide of RNA going in, that would be called RNA polymerase because you're making a polymer of RNA. When you do DNA and you replication, it's called DNA polymerase. That's the added slice that we'll fill in the holes in the next class. So that's pretty much it. What we're going to do is get some computers now and watch some animation.